Hello and welcome to this demo of Serial Cloning. Serial Cloning is an open source web application that you can use to plan your cloning experiments. It can simulate PCR, CRISPR, homologous recombination, Golden Gate, etc. But it can also design primers, it has templates for popular cloning kits, and it can do things like accessing gene sequences from NCBI and much more. Once you're done, you can export your sequences as GenBank or FASTA files, or export the entire history of what you did as a JSON file so that you can revisit it later on or share it with someone. The application is accessible for free at serialcloning.org. Next, I'm going to give a brief introduction about the project, but you can skip it and go directly to the demo. The project has two goals. The first is provide you with an intuitive and flexible web interface to plan your cloning, but also to allow you to export the cloning history in an open format. What do I mean by the cloning history? The series of steps that you follow to go from sequences that already existed to the new sequence you generated in your lab. The problem is that we do not have open data models to capture this information of stepwise cloning. So, with serial cloning, we are developing a data model for cloning history. The principle is very simple. Every sequence comes from a source, and a source can represent either the import of an existing sequence, so for example, a file uploaded by the user, a plasmid from a gene, a sequence retrieved from the NCBI, but a source can also represent an experiment that generates a new sequence, taking some existing sequences as inputs and yielding the output sequence. So for instance, in this ligation, we have that the inputs were the restricted fragments and the output is the final plasmid. So we can see that through a series of sources, we can recapitulate the history of a particular sequence. For instance, this allele that was generated by homologous recombination. We can see that we got a plasmid from a repository, for instance, gene. We amplified a fragment from that plasmid, and we transformed that PCR product into the genome to produce the allele. In turn, the template sequence of the genome came from a genome database, such as the NCBI. So that's it for the introduction. Now I'm going to show a demo that you can follow along in shareyourcloning.org. So this is the website. And let me start by saying that if you're interested in this project, you can subscribe to the newsletter by going to About, Newsletter. So now, how do we start? We need to import some sequences, and we can do this in different ways. We can get some from a repository, for instance, from gene. So we can use the identifier of a plasmid in gene to get it from there. So we just type it here, click on Submits, and we will get the plasmid from gene. Then we can click on this I to see the sequence in more detail. So we can see it as a linear map, a circular map, or see it in more detail in the sequence map here. And we can go back to the cloning tab to import some other sequences. Now let's say we want to get a genome region from NCBI. So we want to get a sequence from the reference genome, and we're going to take it from the fission yeast Schizosaccharomyces pombe. And here we can type the name of the gene, and we will get the identifier from the NCBI. We can click, and then we can request upstream and downstream bases from the locus, so we can see also the region that surrounds our gene. We can click on Submit, and we will eventually get the sequence from the NCBI. So this is the gene sequence, and as before, we can click on the I and see it in more detail. You can also import sequences from files, and you can do it by going here, Submit File, but you can also just simply drop a bunch of files from your computer. So you simply drag them here, and the software will load them independently. So let's for now get rid of these ones, and let's go to a more practical example of cloning. Let's say we want to introduce the coding sequence of this gene into this plasmid. One way in which we can do that is we can amplify the gene with primers that contain restriction sites that are compatible for cloning in this plasmid. So we're going to do that, and for that I've prepared some primers here. If you want to add a primer, you simply click here, you give it a name, give it a sequence, and you can save it. Now let's add a source to this. So this will take this sequence as an input and produce an output. In this case, we want to make a PCR, and the forward primer we want is the ACE1 forward primer that I prepared, and the reverse one is the other one. And we can click on perform PCR, and we see that we have amplified this sequence, but we've added some extra sequences on the side here, and we can see the SAL1 cutting site that we have introduced it, and the ASK1 site that we have introduced on the other side. So we go back to cloning, and we want to restrict this amplified fragment with SAL1 and ASK1. We can perform the restriction, choose the fragment that we want to keep. We of course want to keep the fragment that contains the gene and would leave us these overhangs. And then we do the same with the plasmids. We click on add source, we select restriction, and we use the same restriction enzymes, SAL1 and ASK1. We click on perform restriction, and then we can choose which fragment from the plasmid we want to keep. Initially we have selected this very little cutout sequence that of course we don't want. We want the entire plasmid minus that. So once we select the right one, we click on choose fragments, and then we can click again on add source. Then we want to do a ligation, and you can select the inputs here. So number 10 and number 8 as inputs, and then we can click on submit. Because we only want circular assemblies, we can select this, and we get the final product here. What's nice as well is that you can select a sequence in the child, and it will be highlighted on the parent where it comes from. So you can see that this kanamycin resistant cassette comes from here, and this ACE1 sequence comes from here. You can also just select a random bit, and it will also be displayed in the parent. You can also do this same kind of restriction and ligation cloning in a single step. So instead of first cutting and then ligating, you can do it in a single step. And for that, you can delete those, come back to the initial inputs, and you can select restriction and ligation, which is the same as Golden Gate, really. And then you select the inputs, again, provide the restriction enzymes. We say we only want circular assemblies, and we get the possible outcomes. So of course, one is the same plasmid as before, and the other one is the small cutout of the plasmid ligated to the gene, which of course we don't want. We want this one. So we click on choose fragment, and we get the same product in a single step. So now let's go back to the plasmid and genome sequence that we used before. Let's say that now what we want to do is to delete this gene. So we want to replace it by the kanamycin resistance cassette. This is a typical thing you would do in yeast. And one way in which we can do this is we amplify the kanamycin resistant cassettes through PCR. And in the primers, we add homology arms that are homologous to the sequences flanking the locus, so that through homologous recombination we can replace the locus by the PCR products. You can use serial cloning not only to simulate this, but also to design the primers that you would use. Let's see how to do this. We add a child source to the plasmid, and we want to do a PCR from this plasmid to amplify the resistance cassettes. We can either use primers that we have already designed or click on design primers. If we click here, it will ask us for what we want to do it. So we want to do it for homologous recombination, and it tells us that we have to specify the template sequence. So the sequence where our amplified product will be integrated into through homologous recombination. So in this case, of course, is sequence 4. And once we select this, we can click on design primers. And that takes us to the primer designer. And here we can select which region of the plasmid we want to amplify. So we can see it as a circle as well. And we want to amplify the kanamycin resistance cassettes. Then we can select which region of the genome we want to replace through homologous recombination, and in this case we want to replace the coding sequence of ACE1, so we can select that, but we can also select any arbitrary positions. And also, if instead of replacing we wanted to make an insertion through homologous recombination, we can just select a single position and we can use that as a position for the insertion. Of course, for that we would probably want to go in more detail to select exactly between which two bases we want to do it. But anyway, in this case we want to delete the CDS of ACE1, so we select it and we click on Set from Selection, and finally we come to other settings where we can specify the homology length, the TM that we want, and the minimum hybridization length of the primers that we want, and then we select whether we want the insert to come in forward or reverse orientation. In this case, let's say we want it forward, and we can click on Design Primers, and then the program proposes us with some sequences. So here we have in lower score the homology regions, in upper score the region of the primer that would actually use for the PCR. And if we are happy with this, we can click on Save Primers, and they will be set in the PCR we selected previously. So we can just perform this PCR, which will produce this amplified fragment that we can then use for the homologous recombination here, and obtain our final product in which the coding sequence of ACE1 has been replaced by the kanamycin resistant cassette. We can also download any of these sequences as GenBank files or FASTA files. So we can just click on this arrow in the corner and we can save it as a file in our computer in GenBank or FASTA file, and we click here and we will just save that file. We can also save the entire history of what we've done. So we can come here, click on File, Save Cloning History to File, and that would download a JSON file to our computer. So if we come back to the sites and we either drop the file, it will load it, and you can also do the same by File, Load Cloning History from File. If you are getting started, you can load some examples in here. Click on Examples and click on one of the examples here to load it in the website. Share Your Cloning also has templates that can help you clone in a certain way. For instance, we can go to Templates, Allele Engineering, PCR-based targeting, and we can get a template for what we just did. In this example, we have some pre-populated fields, so we know we want to get something from a genome, and then we also want to get a plasmid, do a PCR from it, and then do homologous recombination. There are also templates for popular cloning kits. For now, there is only MoClo kits, the SIDA and the YTK. Let's look at an example from the YTK. Let's say we wanted to make a self-replicating plasmid with a CDS. The principle of MoClo is that you have these modular parts, and with golden gates, when you restrict the plasmids that come in the kits, you will always generate, for example, promoters that have this particular overhang on the left and this particular overhang on the right, that then you can link to a coding sequence and then to a terminator, etc. So in the template, you can select from the plasmids from the kits, and you can see also what the plasmid contains. So for example, we can select this particular assembly connector, this particular promoter, and so on. And if you wanted to add your own sequence instead of one of the ones that comes with the kit, you can also do that by clicking here. For this example, we're just going to pick some of the sequences that are available from the kit. Once we have selected one for each of them, we can then select the enzyme, which in this case is BSA1. This kit is working with golden gates, and of course we want only the circular assemblies. We click on Submit, and we will get two possible products. So this is the desired plasmid, and the other one is just all the backbones ligated together, which are also compatible, but we want, of course, the one that contains the parts. We click on Choose Fragments, and we can either again download the sequence as a final product, as a GenBank or FASTA file, but we can also download the entire history by clicking here. So that's it. Thanks for watching. If you tried and you're interested, Please feel free to give some feedback or reach out by clicking here, where you can send me an email or create an issue on GitHub. And remember that you can register for the mailing list by clicking on About Newsletter. Thank you and bye.